So that's a new normal, I think. Yeah, cases are increasing because things are opened up. So as expected, it is only going to go up.
It's 10:25 now.
Yes.
Yeah, maybe you can wait a couple of minutes and.
Okay. So, sir, shall we start? Sir?
Why not?
Okay. Okay, very good morning to one and all. My name is Jamin George. I'm teaching at Department of Chemistry, St.Aloysius College, edathua. As we know, this seminar on recent updates on development of vaccine and therapeutics for COVID is one of the most significant topics. And we have with us an eminent scientist, Director Integral Bioscience, Dr.Sreekanth Ramachandran, to deliver this lecture. I now call upon Dr. John, the principal, to welcome.
I think if everybody mutes and only those speaker will unmute, I think you don't have this echo.
No disturbance now? Yes,
Yeah, I will try to mute the mic. All others have muted their mics, actually.
For me, James sound is clear, but I am facing problem with Dr. Sreekanth. I mean, it's a bit cracky sound, it's vibrating.
Okay.
Yeah, I kind of muted now. I think it will be fine if only one can speak at a time. So
okay, yeah, we hope so.
Because we have been also doing a lot of video conferencing, and that's kind of a rule.
Yeah.
Only you unmute it while if you want to speak. That will help.
Okay. So once again, I welcome you all to this webinar. Rajan sir, can you speak now?
Jamin?
Okay. Sir, can you start? Can you speak?
Yeah.
Can you welcome?
Yeah.
Okay.
Yeah. Good morning, dear friends. Respected invited speaker, Dr. Sreekanth Ramachandran, Director, Integral Bioscience, Noida, UP. And dear Dr. Anu, Augustine, Head of the Department of Chemistry, Vice Principal, Srimati Kochudaisyama Joseph, IQAC Coordinator, Dr. Juban Anthony, and NAC Coordinator, Dr. Indilal, and Father Tijomon P. Isaac, and the convener of this program, Dr. Jamin George, and dear participants. The IQAC and the Department of St.Aloysius College, edathua is jointly organizing a national webinar on recent updates on development of vaccines and therapeutics for COVID-19. I congratulate Head of the Department, Dr. Anu, Augustine, and the convener, Sri Jamin, and the entire department and IQAC for conducting such a wonderful national webinar of this kind. As you all know, today we have got an eminent personality to deliver a talk, Dr. Sreekanth Ramachandran. Dr. Sreekanth, let me introduce our college to you in brief. St. Aloysius College, founded in 1965 and managed by St. George Foreign Church, edathua, in edathua Grama Panchayat of Kuttanad Taluk in the Lake District of Alappuzha. The college has now become a well-established, NAC accredited with A grade, with a center offering 9 undergraduate and 5 postgraduate and 2 PhD programs. The college is affiliated to Mahatma Gandhi University, Kottayam. And the college also gained prestigious DST FIST sponsored college tag in 2018. Many things to say about our college, but I stop here. As principal, I welcome you, sir, to our Aloysian family. I wish all the very best for you and for all the personalities behind the smooth conduct of this webinar. Now I invite Head of the Department, Dr. Anu Augustine, to introduce the invited speaker. Over to Dr. Anu Augustine. Thank you.
Thank you, sir. Good morning. I am Dr. Anu Augustine, Faculty, Department of Chemistry, St.Aloysius College. Respected Principal, Dr. Jochen Joseph, today's invited speaker Dr. Sreekanth Ramachandran, IQAC Coordinator, NAC Coordinator, Father Tijo, and all the teachers from our college, and all the dear participants. A very warm good morning to one and all. It's my pleasure and privilege to introduce Dr. Sreekanth Ramachandran, who is the Director of IBS, Integral Biosciences. It's a Noida-based, Delhi-based startup biotech company. So let me introduce himself. So he received his PhD from NIIST, formerly RR Trivandrum. Then he completed his postdoctoral fellowship at the University of Leipzig, Germany. Then he worked at Niger Pharmaceuticals, Edmonton, Canada. Subsequently, he joined the AstraZeneca, that's a British-Swedish multinational pharmaceutical company. It's based in Bangalore, as research scientist, where his work focused on the small molecule drug delivery of anti-infective, mainly in the area of neglected tropical diseases. In 2014, he joined IBS as a lead medicinal chemist, and now he is working as director of IBS. As I already said, IBS is a Delhi-based biotech company which supports medicinal chemistry, in vitro screening, and initial DMB tech characterization of compounds. It's my pleasure to invite my friend Dr. Sreekanth to deliver his talk on recent updates on the development of vaccines and therapeutics for COVID-19. Welcome, Sreekanth. Once again, I welcome all the participants, and I request all the participants to turn off audio and video while the speaker is delivering the lecture. And also one more thing, after the talk, there will be a discussion section. So the participants can either raise their queries at that time or drop it in the drop box. And also, you please submit the feedback form, which will be dropped in the chat box later. Thank you. Welcome, Sreekanth.
Thank you, Dr. Anu, for your kind introduction. And am I audible, right?
Yes.
Okay, thank you. So it's good morning to one and all. And thank you for inviting me to talk about this pandemic, which we are all in the middle. So I have a few slides. So thinking about the audience, I made some general slides and some specific slides regarding COVID-19. So I will start my presentation. So please let me know my screen is visible.
Yes.
Okay. So as you all know, and we all are in this pandemic, COVID-19, right? So I'm just giving you a brief update where we are with the development of vaccines and therapeutics for this pandemic. And so before going into that, and I'm sure people have been reading about this a lot, so I am going to talk about it from a scientist's perspective of this disease, right? I've been working in this area for quite a long time. So I just want to introduce everybody to this disease, first of all. So what is this COVID-19? So it's nothing but it's coronavirus infectious disease. So discovered in 2019. So that's been, it's called COVID-19. And so It is an infectious disease, and it's mainly been caused by this virus called coronavirus 2. So why it's been named coronavirus 2 is that we already had a coronavirus 1 with us some time back. So that was in 2002. So we had first SARS. SARS means severe acute respiratory syndrome, and which is being caused by this coronavirus. So we had the first infection happened in 2002. And then we had another from the same family of coronavirus that's been named like MERS, which is a Middle East respiratory syndrome. In the same family, but its pathogenicity is a little bit different because of the characteristics of the viruses. So that happened in 2012. And now we have a third round of this infection, which we named like SARS-CoV-2, which is coronavirus 2019. So basically, this was first identified in Wuhan, China, in December, and this has been the cause of this severe pandemic across the globe. And it is a group of RNA viruses. I'm going to talk about different sorts of viruses so that people are familiar with how these viruses behave and what are the way forward, and which causes severe respiratory tract infection, which can range from mild to lethal. Right, and this is a zoonotic infection. That means it was originated from animals, and which was later transmitted to human beings, which happens in a very long-term thing. So all these are zoonotic infections. So it's been transmitted from animal to human, either by contact with animals, mostly by continuous contact with animals. So the origin of SARS-CoV-1 seems to be some Chinese cats which have been preying on these bats. So mainly all these are coming from bats, but there are animals who eat the bat, they get infected, and then people who are carrying these as animals as a pets or for meats, they're getting infected, and that's how that human transmission is happening. So, and we all know these numbers. We are always doing this number business every day, right? Infections in India. So you can see. So this number I have taken from yesterday's. So basically, there is active cases of around 3.3 million across the globe, and there was 4.2 have closed cases with a mortality rate of around 10%, with around 424,000 people died of this infection. So this is a very high mortality rate for an infection like flu, which initially believed to be a flu-like thing, right? And we all catches flu. So these are the symptoms, which the major symptom is fever, cough, loss of appetite. Is similar to all the viral infections, which we are familiar with. But if untreated and if you have comorbidity, then it becomes lethal, especially on elderlies. So I'm just going to talk about a little bit about this viral infection in general. So, viruses are the small infectious organisms, it's microorganisms which can't be seen by naked eyes. And, but they don't survive as such. They need a host to survive. So, it comes from when an infected person to another person. So it is transmitted mainly by air and the particles, by coughing and sneezing. So when a person gets infected by being close to a person who is infected, such as by cough or sneeze, so it travels through our mouth, nose, and eyes, basically, into our body, and it gets into the host cells. It enters into the host cells, and then it starts replicating. So by themselves, they don't survive. So if a virus comes in on contact with surfaces which there is no host particles, it will stay for some time and they dies off. So they survive on host cells. And there are two types of viruses so far been known. They are called DNA viruses and RNA viruses. So they are basically made up of these genomics, whether there is a DNA virus or RNA virus. But all this coronavirus family belongs to RNA virus. They are made up of RNA. So, such as HIV, Ebola, SARS, COVID-19, common flu, influenza. So these are all RNA viruses, and they are very prone to mutate as well. So if you look at the lethality of these infections, basically these RNA viruses. So these are some of those numbers you can see. So we have heard about Ebola. So it's not very contagious, but it's highly lethal. So if you look at the fatality rate, it's 50%. So half of the people die of this infection, Ebola, which mainly happened in African, sub-Saharan countries. And then you have this Middle East syndrome, which also is highly lethal, around 35% of fatality rate. And then we heard about smallpoxes, which now we have a very good vaccine. So that's also was very lethal at the time when it was prevalent. And then we have SARS-1. This number is for SARS-1, which was around 10%. And this was taken some time back. So we are, this final number for COVID-19 is not there because it's still ongoing. But right now it's around 3 to 10%, right? But we'll only know when the first wave is completed, really, because there are a lot of active cases still ongoing, right? So these are the numbers. So 10 means it's big, because if you look at our seasonal flu, it's only 0.1, right? And we are getting seasonal flu every year, like 2 to 3 times, and we are not dying of seasonal flu. And so this, when compared to seasonal flu, this is much more lethal. So that's a reason people should take protection, right? And right now, we don't have good ways of protection. That means we don't have a vaccine, we don't have a good therapeutics. So ways of protecting, diagnosing and treating viral infections such as COVID-19. So once you have a vaccine, you can protect it. But before that, what we need to do? Okay, so once we just came to know about a new virus, the best protection is by social distancing, physical barriers, and try not to get it in first place. And then we have our body's immune systems, which can attack the virus. But I will talk about the immune system in a following slide. There are 2 types of immune systems in our body. And then how you diagnose this sort of infection. So basically the gold standard diagnostics is by nasal or throat swab using a PCR technique. It's a lengthy process. It's a time-consuming process. That technique try to detect this RNA virus in the swab particles in the swab test. So it usually takes 1 to 2 days. That's a turnaround time. And then you have this faster rapid diagnostic kits which checks for the antibody. But it's not a confirmatory test. It's just a first test. And if you are positive on the antibody test, you should get a PCR done, which will confirm that you are either infected, because the antibody will only tell you have the antibody developed, right? So it can happen after infection as well. This antibody tend to stay in your body for some time. So antibody test doesn't tell you that you are currently infected. It will only say that you have been infected with this virus, whether you are likely infected or in the past. So PCR test is the gold standard, and then comes about treating this. The protectives to treat these viral infections are basically vaccines and prophylaxis drugs. So and I will just talk about different types of vaccines and what is happening in this area as well. And then we have antiviral drug available for other viruses. So, but the discovery for these are in progress, right, for this particular disease. And then we have immunosuppressive therapy. This is something new for this disease, right? Most of the flu viruses, influenza and other things, we don't need this immunosuppressive therapy. So what's happening with this particular virus is that in some people, when it gets infected, our body try to protect it, right, against a foreign particle. So it releases a lot of cytokines. Our protective mechanism is called cytokines. So it creates a lot of cytokines in our body just to push off this virus, kills the virus. But this that cytokines overwhelm our body, right? And even too much of cytokines is also bad for us. So, but the body's only way to respond is by releasing those. So people die of this cytokine. It's called cytokine release syndrome. So some people die not because of this virus, but our body's defensive mechanisms. So that's why in hospitals, doctors are treating both with antiviral drugs and immunosuppressive therapy, because you have to balance this both on critical care patients. And something which is come up, which is not a real cure, is plasma therapy, which is because for patients who are cured of this disease, they still have the antibodies generated against this virus. So plasma from the cured patient can be used to treat another patient, so that these antibodies are transferred into that patient so that they can be infected. So this is also being tried because we don't have good vaccines and antiviral drugs. So this is what it is happening right now. And let's look at the potential, cure in this area. So for any particular disease, so there is a lot of basic research is needed, right? Try to understand this virus. Okay, is it similar to the previously known things? And then only you can use that as a similar drug, right? If it is very distinct from a previously known virus, then you have to do a lot of basic research. Try to understand its genome, right, and its sequences and its drug targets. Where to target this drug using small molecules or antibodies or vaccines. So you have to do a lot of basic research in order to understand a new virus. And then I talked about the diagnostic part. Even in diagnostics, we need some more innovation, right? We can't wait for 2 days to get a test results. And if that test results somehow become, not very reliable, you need an improved diagnostic thing. So there is a lot more research is happening in this diagnostic area as well. A lot of Indian companies are also doing, a lot of companies globally also working on this for a rapid, more reliable test. So I just recently heard there is something called saliva test been approved in US. So you can take your saliva at home and send it to a lab and they can test it for virus. So these are the kind of innovations happening in this diagnostic area to get a more rapid, more easily accessible test for this particular disease. And then we have vaccines, which is the protective mechanisms. That need to be developed, right? And there is a lot of R&D going on and it's a lengthy process. Usually these are all lengthy process, but here we don't have time and we are running against a race, right? So currently they are testing some of vaccines, newly developed old vaccines against this particular disease in human clinical trials. And you need to do some, animal studies before that. So just to look at the safety of those things before you go into human trials. So that's been happening here. And then you have a therapeutics to target this virus itself. So there are different ways of doing it, repurposing known drugs. And I told about immune therapies because there is a lot of immune activation happening in our life. And people are also trying novel compounds to treat this particular disease. So these are the nutshell thing. And one thing I want to just talk about is that why this virus is different than other viruses and why is it causing so much fatality, right? So people are trying to understand it. So there are a lot of studies coming up every day. So basically, when it gets infected, when this virus got infected, I told you it attached itself to a host cell. All right, that host cell's name is called ACE2. It's called an enzyme, angiotensin converting enzyme. So in our body, we have a lot of this ACE2. And from people to people, the percentage of this enzyme varies. Right, and this could be the reason, one of the reasons why some people gets infected a lot, or it becomes severe, and some people get a very low level of infection. So one of the reasons is that this particular enzyme in our body, which is this virus get attached to that enzyme, and then it enters into the cell, and then it replicates. And this is most abundant in our lungs. This particular enzyme is most abundant in lungs. So that's why you start getting this inflammation and coughing, shortness of breath, which is all related to lung, right? And so this, so there are a lot of studies happening here along this, looking at the levels of this receptor from different type of people, right? It matters from moving from ethnicity, moving from Asian to Europe to Americans or Africans. So there are a lot of variability in ethnicity, and there are gender differences, right? And there are a lot of studies even in blood grouping, right? We all heard about this lot of differences. So there are a lot of studies happening why some people get low infection or asymptomatic in that sense, and why some people are getting severely infected. So one of the reasons is because of this ACE2. And associated with that, this body's mechanism of releasing cytokines against this virus. So our body's immune system generates any foreign particle, against any foreign particle, whether it's virus or bacteria. the basic killing mechanism from our body is the cytokines. And there are many cytokines known in our body, such as IL-2, IL-7, IL-16, and these are being released so that they go and kill this virus. But sometimes these overwhelms in our body, and this also become a cause for mortality. So this is some of the pathogenicity, and something more about this ACE2. So as I told, this is highly expressed in lung, and it also has a role in cardiovascular and immune systems. And it involved in heart function, and that's why you might heard that people having hypertension and diabetic, they have a high expression of ACE2 in their body. So that's the reason probably you see a lot of mobility in that area. People having hypertension and diabetic, they already have a high level of ACE2, so they are getting a lot more, severe because they have a lot more ACE2. And there is one thing also I read is that smokers also have high levels of ACE2. So that is that's also been studied, right? So smokers might have a chance to get it more infected. So how it binds is that so if you look at these pictures of this corona, they have a crown-like structure. So they are called spike proteins. So that spike protein attaches itself to ACE2, and because of the strength of this spike protein, this particular virus is behaving a little bit smart. When we consider the previously known coronavirus, their spike protein was not that big. So one of the studies shows that because of this spike protein, this can attach more strongly to our host cells, and that's how this is being getting lethal. So there are a lot of studies happening on this ACE2 and its relation with this coronavirus. So yeah, like I told, cigarette smoke accumulate large number of ACE2 receptors. So smokers are probably more prone to this. More prone means that the severity might be more for those people when compared to non-smokers. So that's one of the studies being done. So yeah, let's talk about the human immune system. Right, we all know that we talk roughly about the immunity, right? Are we immune? Right, there are two types of immunity in our body. First is called innate immunity. That is the non-specific defense mechanism of our body, right? That means when a virus comes into the body, are we ready to fight it off? Plus, we have physical barriers such as skins, these immune cells in the blood, etc., etc. So that's a first level mechanism for our body. Whenever we get a foreign particle, this innate mechanism works. And then there is called adaptive immunity. That means for a particular organisms or a foreign body, our body tend to create agents such as antibodies. Okay, these are special agents specially made for that particular thing. So these are very specific to each pathogen. So this is called adaptive immunity, but it takes time, right? When a foreign organism comes, and this is more protective rather than this innate mechanism. So this adaptive immunity takes time. It takes about 2 to 8 weeks to create these antibodies against a particular pathogen. So the vaccine being is based on this adaptive immunity mechanism. So, but it takes time. So that's the difference between innate immunity and adaptive immunity. So, and for this particular virus, mind you, none of us have this adaptive immunity at this point. We have some sort of innate immunity, probably that's not enough to fight this infection. So that's the reason everybody should be careful, right? Everybody should be masked. People should, if you were to go to a crowd, everybody should be masked. Because there are a lot of asymptomatic people out there, right? We don't have this adaptive immunity for this particular virus right now. And we don't know how much severe it could be once we get infected, right? Otherwise, there is a scenario called, like, you might also heard about herd immunity. Herd immunity is nothing but 50% of the entire population getting infected, at least 50%. Then they place like a barrier for the 50% of people. So we are not into that level right now. To get into that 50%, country like India, we are 1.3 billion population. So at least half of the 600 odd million people need to get infected to get into that level. So that's a long way off. So don't be thinking that this virus is gone and I am free of this virus. So because none of us have this adaptive immunity against this particular virus at this point. So it's the only way to protect ourselves is social distancing, wearing mask, and proper hand washing and sanitizing, right? And there is a race against to develop vaccines. So vaccines is based on the same philosophy like adaptive immunity. So this is an artificially created immunity in our body. And so I told, for each pathogen, the antibodies are different. So you probably can't use an antibody which is developed for a different pathogen for this one. So you probably are ending up to develop a new one, right? And to develop a new one, there are a lot of hurdles. A vaccine basically contains key structural or functional components of this pathogen, which stimulates the immune system to produce antibodies and immune cells. So that's where the basic research comes into. So let me talk about the different types of vaccines, which has been known and been tried out successfully. There are 4 types of vaccines available for us for different infectious diseases. The first one is a live attenuated vaccine, which is a live vaccine using a weakened form of this disease. All right, the virus or one can tweak the virus to make it weakened or attenuated. Not a live virus, but attenuated virus that can be used like a vaccine. So we have examples of measles, chickenpox, rotaviruses, yellow fever vaccines available, which are these live attenuated vaccines. So you generate antibodies, your body generate antibodies against these particular diseases using these live vaccines. And then you have these inactivated vaccines. So these are use a killed version of the germ that causes disease. So this is probably not fully killed, the live vaccines. So there is a fully killed vaccine, which is called the inactivated vaccine. So that's been employed so far for hepatitis A, flu, polio, rabies, etc. And there is another type of vaccine, which is a recombinant vaccine or a conjugate vaccine. So these pieces, they use specific pieces of this germ, like its protein, its RNA, or its components. So for this particular COVID-19, I think people are engaging more into this category, and there are some attempts on these levels as well, but more work is going on in this third category of viruses. So, but let me talk about the challenges as well. It's not as easy as we talk, because there are a lot of scientists working across the globe on this challenging problem against time. Right? Development of an effective vaccine is a challenge which is typically guided by decades of basic research in viral biology and host response to infection. So for a very successful vaccine, it usually takes 10 to 12 years because you have to do a lot of basic research on that, both in terms of efficacy of this virus vaccine and against the safety of the people who are taking these vaccines. So against the backdrop of these many unknowns in this current scenario, especially in the coronavirus, there are some risks associated with expedited vaccine development, right? So that's again a problem, right? We have to face, because everybody wants a vaccine now, but there is no easy way of making it. So people are trying to test it out in a small population and trying to find the effectiveness, but only when it's been engaged in a bigger number, we will really know the effectiveness, because I told you the ethnicity matters, the gender matters. There are a lot of variability in terms of our protective mechanisms. So for that, you need a broader clinical trial, which takes time, and we don't have time. Right? So the first generation vaccines which might come out probably early next year, let's say, it may not be the best vaccine available for this, but that's where we are right now, because of the pace we need this vaccine. So while a gold standard vaccine will take some time, the contest is making about about getting some prestige in developing this, also saving millions of lives and money. Everything is a business, and into this development of vaccines. So for all these companies are putting a lot of money into this development process, and they need to get back some to sustain their business, right? So let's talk about the stages of vaccines development. And there is two components, a traditional way of making it and in a pandemic mode. right? So I told in a traditional way, it takes a longer time, right? 10 to 12 years. So because that involves a very discovery level research, which happens in the labs. There are a lot of research labs in India and abroad, right? So they look at the virology, the antigen identification, expression systems, immune assays. There are a lot of in vitro assays. That means the assays which are being done in the test tubes or petri dish. So that's been tried out in the labs to see that whether this synthetically made vaccine work against this virus in a test tube. Right? If that is been successful, they go for scaling it up a little bit more and try to do that vaccine in animals. So there is a preclinical animal studies required to test it out, the success of this vaccine, because this animal will be pre-infected with this particular virus, or they'll be treated with a vaccine first and they'll be infected then. And see levels, they can check for the levels of antibody being generated in animals and see whether they are protective. The animals are protective against this virus. So there's a lot of animal tests required before moving into human clinical trials. So if this is successful, again, this might take 1 year time, 1 to 2 years for success of animal testing. And then it goes for human clinical trials. So even in human trials, there are different phases. There are phase 1, phase 2, and phase 3. So basically in phase 1, you check for the safety of this vaccine in humans. So this vaccine should not be toxic to healthy people, right? So healthy volunteers will be involved in phase 1 and they look for the safety and the doses which are required to create the antibody, right? You can increase the dose and see that are you getting enough antibodies in our system to protect it against this virus. So there is a dose escalation happening in phase 1. And then in phase 2, you look at a smaller people where there's a prevalence of this disease, right? You go to community and try to treat people on a smaller number of people on phase 2 and look for the efficacy. If that has been successful, then they push for a bigger population with a diverse ethnicity. They go for thousands of people. So their phase 2 will be hundreds of people, and in phase 3 you have tens and thousands of people being tested out. So again, this process will, in a traditional mode, it could take something like 3 to 5 years. And then it has been successful, then it submits for approval, right? There is a lot of approving agencies. There is FDA in US, and there are other regulatory authorities in India looking at for approval, and they look at all the documents and its efficacy, and then they license it out one particular company. And then there is a manufacturing process. So during this phase, they will try to scale it up. In a manufacturing plant, right? You need to treat it to millions of people, right? So the scalability also matters. So from a small scale, you have to go up into really big scales of treating the people, right? So then it goes into production and then it comes into market. So, but in a pandemic mode, you don't have the luxury of doing all these things, right? And because you need vaccines as soon as possible. So what we are trying to do is right now is looking for a shorter time period, like in a year. Can we get a vaccine? Because this first happened in December 2019, right? And now we kind of covered like 6-7 months now. And so what people are trying to do, they're trying to use off-the-shelf existing vaccines plus some very fundamental tweaking of something known. And they're trying to do some animal work, but little because they don't have time. So they are skipping animal work. So there's a risk, right? Skipping animal work means that there is a risk in terms of safety and efficacy going into human trials. So, but in a pandemic mode, that happens. So people are pushing some vaccines into this phase of phase 1 and 2 clinical trials, and then quickly moving into phase 3. So that's why there is a risk associated moving into this phase. We might end up with a suboptimal vaccine or an unsafe vaccine. So that's why we are, because we are in a pandemic mode, right? So this is the status of vaccine development, and